get started. Okay, so today is Chapter 5 of Janeway, the generation of lymphocyte antigen receptors. Okay, so the lymphocyte antigen receptors are how the lymphocyte sends antigen in the environment, and on B cells, this is actually just IgM or immunoglobulin, which is also the B cell receptor, and T cells actually have a specialized T cell receptor. But each receptor has a lot of differences that make a wide range of specificities, and this is primarily due to variation in the V region, and it's in the amino acid sequence of the V region. But of course, the amino acids are translated from nucleotide sequences, which means that the nucleotide sequence has to vary on the DNA before the RNA is even transcribed to bring all this, this variation. So the V region is encoded on several chunks of DNA, several chunks of nucleotides on the DNA called gene segments, and then they are reassembled using something called somatic DNA recombination or gene rearrangement. So somatic, remember somatic means not from the gametes, so the gametes are haploid, so it's the ovum and the sperm. Somatic are all the other cells outside of those. This is happening in other cells, namely the lymphocytes, which are diploid. They have 2 copies of DNA on them. And there's actually gene rearrangement going on here. That's what the somatic means. So once this has happened during lymphocyte generation, you get an entire repertoire of different antibodies. So B cells, this is happening in the bone marrow, and you're generating lots of B cells. And each one is going to have a receptor or an IgM on the outside that is specific for something else. And all of these different specificities together are going to make up the antibody repertoire, which is, we estimate, about 100 billion different specificities. Extremely, extremely broad and diverse. Okay, so how does this all come about? Well, like I said before, it starts with the variable region being in gene segments, right? So traditionally, if you're going to transcribe a gene, you're going to have a bunch of nucleotides or a bunch of letters, and the transcription is just going to come straight across to make RNA, which is then translated to amino acid based on those 3 nucleotide codons. It's just read straight across. But the variable region of these immunoglobulin receptors are actually in these gene segments or chunks. And we divide them into 4 different categories. The variable region anyway in 3 different categories. So there's the V segments, the J segments, and the D segments. The D segments, however, are just on the heavy chain. On the light chain, it's just V and J. And then both have a constant region that has a different length. So in the light chain, what has to happen is that you take one of many V chains and combine it with one of many J chains, which then gets combined with a constant region. And then you have something that can be transcribed into RNA, recombined to be close together in one nice sequence. The heavy chain, you have to rearrange a D and a J, and then a V, a D, and a J. And then you have something that can be transcribed, variable region, and then connected immediately to a constant region. Okay? So this is the first part of diversity on the immunoglobulins. because you have lots and lots of different segments that can be combined. So you have remember there's two different types of light chains, the kappa and lambda light chains. Kappa light chains, you have somewhere around 35 different variable regions. Lambda, about 30 different. And then on heavy chains, you have potentially over 40 different variable regions. And in heavy chains, these 40 can be combined with any one of 23 different diversity regions. And then that can be combined with any one of 6 different joining regions. On the light chains, the V can be then combined with 4 or 5 different joining regions. So you can see if you can multiply all the different permeations that could happen, you can just from this alone generate an incredible amount of diversity. So all this is happening on different chromosomes. So the lambda light chain is encoded on chromosome 22, kappa on chromosome 2. The heavy chain is encoded on chromosome 14. If you haven't already, a list that's probably reasonable to start putting together are the different locations of all the different parts of the immune system on chromosomes. Probably one of those lists that you would just memorize quickly before the test. I think maybe we've talked about the MHC so far. And now we're talking about the immunoglobulin receptors. On the different chromosomes, on the different loci, this is kind of what it looks like, right? So we said lambda has something around 30 different variable regions, so they're all lined up. Then you have some J regions that are all lined up that you can use. Kappa, very similar. Variable regions, J regions, and the heavy chain has the D. So there's a bunch of different V options, bunch of different D options, bunch of different J options. You need to pick, for example, in the heavy chain, any one of these Ds to randomly combine with any one of these Js. You get a DJ rearrangement, and then you have to connect it to a V, and any one of these Vs can randomly connect to this DJ. One of the first big points in this chapter are things to memorize. That in heavy chain, the DJ rearrangement occurs before the VDJ rearrangement. I remember this by remembering that DJs or disc jockeys came before VJs, or video jockeys. DJ before VJ. In light chain, it's just V to J, and that's all that happens. But here, DJ and then VDJ. Light chains, VJ. Okay, so what signaling occurs to know this happens? Well, as it turns out, there's something called the recombination signal sequence, or RSS, that occurs right near each and every one of these different gene segments. So an RSS is a specialized region that starts with this heptamer, which is CACA-GTG, and then has somewhere between 12 and 23 nucleotide sequence. And then it has a nonomer, ACAAAACC. So this all together is called the RSS, the recombination signal sequence. And recombination has to occur between RSSs of a 23 and a 12. So kind of complementary. So you don't get a 12 and 12. You get a 23 and 12. So this V combines with J, V combines with J. And here, D combines with J, and V then combines with VJ. But this is 23 and 23, so on the heavy chain, the V's can't recombine with the J's. Okay? So I may do an aside if there's time at the end to look at some sequences, just to get comfortable with looking at sequences. Remember, all you're thinking about here in DNA is a whole bunch of letters strung together. In some place, there's going to be CACA-GTG, nucleotide times 23, ACAAAACC, and that is going to be a signal that we can recombine here. That's going to be this RSS. So how does this happen? So for whatever reason, randomly a V is chosen to combine with some random J. So the two are going to line up next to each other most of the time. And then this is actually going to, this loop that's created is going to get cut off. And then this DNA is going to get repaired, making this V and J close together. And then everything in between is just going to get deleted and sent out. So in B cells, so in immunoglobulin, this is something called a CREC, K-R-E-C, which stands for something I never remember, but I think I wrote down. Oh, maybe it's kappa-associated recombination excision circle. In a T cell, that circle that's deleted is what? TREC. T cell receptor excision circle. Maybe I didn't write it down. Oh. Anyway, in B cells, it's called a CREC, and it's not as readily being used yet as TREC to see if you've actually generated T cells. But I think people are experimenting with it to see if you can actually generate B cells and to see what the clinical utility for it would be. So sometimes you can actually get a different type. So this is kind of the standard to remember, but just as an FYI, sometimes what can happen is you can actually get a loop where it's all reassembled into one piece. So it kind of turns on itself, or let's see, there's a term for this, inversion. It inverts and then recombines in two spots. So everything is preserved in the sequence, but you still actually get a V and a J that come back together in close proximity, so you can then transcribe. But everything else ends up downstream together. But the vast majority of the paradigm we're going to learn about is this excision. You're going to get an excision out of here. So how does this happen? Okay, so again, just randomly NEV and NDJ is going to have this 1223 RSS. One's going to have 12, one's going to have 23. They get lined up. They get lined up by RAG. RAG is a complex made of RAG1 and RAG2, which probably everyone's familiar with from a form of severe combined immune deficiency or Omen's syndrome, or we're finding it in granulomatous disease in older kids, different mutations in it. The RAG is actually going to take hold of that RSS, combine it with the J in the RSS, and it's going to hold it in close proximity, and then it is going to cleave the RSSs and break off a piece, and it's going to close the DNA that's going to be preserved with a hairpin. So this hairpin is this tiny loop right here. So it's closed by a hairpin. You can see it here. This little hairpin closed. Okay, so RAG has cleaved it and excised out this join or this CREC. In the case of the CREC, it's very easy. It's going to bind to something called Ku70, Ku80. It's going to get bound, and then DNA repair enzymes that are ubiquitous, that are in all different cells. They're not specific for lymphocytes, are going to come along and just repair it, and you're going to have the signal that's just kind of waste product. But we can exploit and find in the blood. But to rejoin, okay, so this RAG, this is specific to lymphocytes. Now you have broken DNA, and DNA can break, right? Like do things like radiation. So in all your cells, you have enzymes that can repair DNA. So then this process takes hold because now you have broken DNA. So what happens? Something called Ku70, Ku80 come by, and it grabs both ends. And then a complex made of DNA, PK, and Artemis will open up these hairpins. And then something called TDT processes the ends, and it processes by adding nucleotides or N-nucleotides, which happens to add variation at the same time. And then the repair enzyme, DNA ligase, comes through and hooks them back together. But this whole scenario is imprecise. It leaves a whole bunch of variation here where the V was and the J was. A bunch of nucleotides are left in there, which is good. We want this variation. So we have some combinatorial variation in that a random V and a random J just came together. And then you have some junctional variation here from this imprecise joining when a bunch of nucleotides were added. Okay? Does that kind of make sense? This is the big concept. This is, I think, out of everything, if you learn nothing else from this chapter, this slide, you should know cold by the end of your fellowship. Rag brings them together, clips it, closes the hairpins. Artemis, DNA PK, opens those hairpins. TDT adds nucleotides. Ligase puts them back together. This is just a closer look. I'm going to repeat this many times. Rag grabs the RSS, brings them together, clips it off, closes the hairpins, tiny little hairpins. K70 and 80 binds the DNA ends. DNA PK Artemis complex opens up the hairpins, allows TDT to add nucleotides, and then DNA ligase 4 can put them back together. It all goes away, and you have a recombined DNA segment that can be translated. Okay. So, like I mentioned before, when antibodies rearrange, a lot of diversity is made. So the combinatorial diversity comes from the random different V, D, and J segments that are put together. Junctional diversity occurs from this process we just looked at, all these extra nucleotides that are added when the DNA is repaired. Then you also get some diversity because the different heavy chain and light chains that are made in each cell can then be combined. And lots of different heavy chains could be made, and lots of different light chains could be made. And a heavy chain and light chain is going to go together to become an antibody receptor. And then specific to antibodies, it doesn't happen in T-cell receptors. You get something called somatic hypermutation that we'll talk more about in a minute. Then you end up with some regions that are just small areas of very high variability, but happen to land in the groove of the antibody where the antigen is actually going to bind. Remember, we call these complementary determining regions CDR1 and 2, which occurs at on the variable, the V region. And then there's something, the CDR3 has the highest area of variability. And this, I believe, occurs at that junction between the V and J. Yeah, CDR3 falls at the joint between the V and J segments. There's going to be a lot of variability. CDR3, commit that to memory, is the most variable. It's at that junction. CDR1 and CDR2 are at just on the V region. So when this reconnection happens, there's actually two different sets of nucleotides that get created. One set is called p-nucleotides, or palindromic sequences. So it's a palindrome. Palindrome is something that can be read backwards and forwards, like dad is a palindrome. No matter which way you write, if you read it backwards or forwards, it's the same word. Right, so this is similar to the p-nucleotides, and I'll show you that in a second. They're palindromes. I'll show you why they're palindromes. So N-nucleotides are non-template encoded. They're added by this TDT, up to 20 of them, as the ends are being cleaned up right before the DNA ligase puts them back together. This is actually less common in the light chain genes and more common in the heavy chain genes. I'm not sure that's an important point. But the P and N nucleotides together add a whole bunch of diversity at that joining region. So where do these come from? So remember the RSSs are brought together, and here's the RSS. If we take a closer look, remember what I said they were, CAC, TG, TG, TA, is that right? CACAGTG, CACATGT, right? So here is your heptamer. CACAGTG. Your two heptamers of your RSSs. They're in close sequence here, right? And then RAD comes along, and it's going to clip these off, and it creates this hairpin. Okay, now remember, in DNA, you have ACTG, right? And A's always combine with T's, and G's always combine with C's. So Artemis is going to come along, and it's going to open this hairpin, but it opens this hairpin. It's actually going to flip it open like this. So then you have TCGA. These are flipped open, TCGA. Same here, ATAT. It's going to flip it open. So what's happened is you've created a palindrome. So this reads ATAT. But then if you were to look at the bases that should be complementary here, ATAT. Here you have TCGA, but what combines with A is T, G is C, C is G, and A. So no matter which way you would read it, it'd be the same if it had that second strand. So it's a palindrome. But then this TDT comes along, and it just randomly is going to add some nucleotides. And you're going to fill in the complementary nucleotides. And it's going to be ligated. So you're going to end up with parts of these palindromes, which may not be real obvious anymore as things got cleaned up. And then you're going to have just these random N nucleotides. So you're going to have P and N nucleotides in this gap. Now the T cell receptor rearrangement is extremely similar. So think of the alpha chain on the T cell receptor like a light chain, and the beta chain is more like a heavy chain. So you're going to get an alpha chain generated and a beta chain generated, and then an alpha and a beta is going to combine, and you're going to have a T cell receptor with a high amount of variability generated from basically the same ways. So in alpha, you're going to have a whole bunch of V segments, a whole bunch of J segments, a random V and J. So you're going to have some combinatorial diversity. The beta chain has a D segment like heavy, so you're going to get BDJ rearrangement, and you're going to get a whole bunch of combinatorial diversity. So right, you have your variable regions, your J. You're going to have a short constant region on alpha. Beta chain, you're actually also going to have these D regions. So you're going to get on the alpha chain a VJ recombination event that creates an alpha chain. Beta chain, you're going to get a VDJ, DJ first, and then VDJ rearrangement, and then you're going to have functional alpha and beta chain, which are going to combine to make your T cell receptor with a high amount of variability. You have your rearranged DNA. It can be transcribed into RNA and then translated. The codons are translated into a protein that will fold into a T cell receptor. Interestingly, remember there's not only alpha beta T cells, although this is upwards of 95 or more % of all T cells in the body, but then there's also these gamma delta T cells. So this is just one interesting tidbit that may be worth memorizing, that the delta locus, I'm just skip ahead and come back. The delta locus is actually found within the alpha chain. So once the alpha recombines, it actually excises out all the delta. So then a T cell, we think, has potential to either become alpha beta or gamma delta. But once an alpha rearrangement has occurred, all the delta genes are gone, so it cannot then become a gamma delta T chain, T cell receptor or T cell. It's one of the other because that delta is contained within the sequence. Here I think it is. Here you have your alpha chain, but then right in the middle of that you have your delta chain. Gamma is completely separate. So this is just a look at the different variability that you can get here. So you have about 40 different variable segments on the immunoglobulin heavy chain. Kappa plus lambda together, you get about 70. You get 23 diversity segments of 40 times 23 times 6 different joining. With the addition of P and D nucleotides, so you get an amazing number of total diversity. This is very similar in you have a huge number of variable segments in both the beta chain and the alpha chain. You have diversity segments in beta. You have different joining segments, much more in the T cell receptor. You still get addition of P and N nucleotides. An alpha beta T cell receptor recombines as well. So all this together, the junctional and combinatorial diversity leads to just an amazing amount of total diversity, making up that whole antibody repertoire and then your whole T cell receptor repertoire. There is no, however, inherent differences by the way in the way the T cell receptor occurs. It's using these same mechanisms. RAG, Artemis, PK, to splice out the DNA and recombine it. So two things can happen in the immunoglobulin, receptor that does not happen in the T cell receptor. So one is something called class switching, where the activated B cells switch the constant region, that heavy chain. So it always starts as IgM and IgD. So IgM and IgD are right there. And that happens because the IgM and IgD are actually the next in line. Once you've had your VDJ rearrangement, and it is VDJ because it's a heavy chain, you have VDJ. Just by default, M and D are there. And then further downstream, you can change to GE or A. But you actually have to return on these mechanisms and cause another recombination, DNA recombination event to get a class switch. This is controlled by different signaling. So the heavy chains, there are 9 different heavy chains. So there's just one IgM, and we usually designate them, the heavy chain, by their Greek letter equivalent. So IgM is the mu. There's one D and one E, and then there's 2 different IgAs and 4 different IgGs. Sometimes people talk about checking IgG subclasses. We're not really sure what the clinical utility is. But 4 different IgG subclasses, 2 IgA, an IgM, IgD, and IgE. So there's 9 different things that can be can occur in the switching. There's 9 different options that can result. And each of these, like we've mentioned before, have different properties. So some are very, very good at activating complement, like IgM and IgG, particularly 3, 1, 2. Some, like IgG, 1 and 3, are very good at transferring across the placenta. IgE, of course, has a high affinity for mast cells and basophils. So it is advantageous to switch and to turn on that new constant region, that new heavy chain, in order to diversify not what the immunoglobulin can bind to, but then the effector function of the immunoglobulin. Like I mentioned before, right, on the human DNA, you have your VDJ, but then right after the J, you're going to have mu and delta. And this is the only time you can actually have 2 different classes of immunoglobulin on the same B cell. So once it's switched, in general it's switched, and that's it. But it's actually it's an RNA event. It's not a DNA event. So an RNA modifications you can actually get in the same cell immunodelta. So on one B cell, you can have both IgM and IgD. We don't talk about IgD that much. So don't completely take it out of your mind. The rest, actually, it takes a DNA recombination event to then splice out part of this. So if you wanted to reach IgE, you have to splice out all of this. Even after the B cell, it always starts with IgM, but then when you find this variable region is very good, then it's advantageous for that to switch maybe to a plasma cell and start secreting IgE. So you want to splice out all of this class switch. So some of those mechanisms are turned back on, this is taken out, it's switched to IgE. So the structure of the immunoglobulin, depending on class, is very similar, except just take note that IgD, G, and A only have 3 segments of constant regions, or 3 immunoglobulin-like domains here. IgE and IgM have 4, but then lack this hinge region. So D, G, and A have this nice hinge region, whereas E and M is substituted for a 4th immunoglobulin-like domain, or a 4th constant region on this heavy chain. Right, so here's what I mentioned before, IgM and IgD. So there is only one instance where the same B cell can make 2 different immunoglobulins, and that's right at the beginning when the B cell is first made, before it's encountered antigen. It always starts with IgM and IgD. But that's not because the DNA has changed. The DNA is always going to be transcribed the same into RNA, but then it's RNA modification, it's RNA processing that has the choice here of where it's going to end. So it can actually end within IgD tail constant region, or it can pick up mu as IgM. It can do either, but this is not at the DNA level, this is at the RNA level. So this is a little bit different. So you can have both IgM and IgD. So the second modification that can occur, that does not occur in DNA, is that any immunoglobulin can come in a transmembrane form or a secreted form. Transmembrane form has this short membrane region. But again, this is at the RNA level, which can be spliced out. So it can come in 2 different forms. Now, this is not, a unique concept, right? So any gene can have various transcripts depending on which exons are going to get spliced out. Some can actually be transcribed in different directions. This is no different. So don't let this, confuse you and think it's this big unique concept. It's not. It happens a lot in the body, in DNA. The big difference is in this DNA rearrangement. But once that gene is made in that cell, there, and then this is all kind of post-processing. So you can splice out this transmembrane region, and you can either have secreted immunoglobulin or transmembrane connected. It can be the same even if this was IgG. You can have secreted IgG, or you can have a B cell with IgG receptor on it. Once it's secreted, what's happened? The B cells will declare it and it's become a plasma cell, and now it's secreted. Once these are secreted, 2 have a specific property of dimerizing, polymerizing, right? So IgA gets combined into a dimer. IgM, once it's secreted, becomes a pentamer, and this all happens with the assistance of something called a J chain. The J chain changes an IgM to a dimer, and a J chain helps an IgM become a pentamer. So this becomes a very, very big immunoglobulin. But we've talked about all this before. I'm not going to spend much time on this, but remember, it's a J chain that does this. So I'm more concerned about spending the rest of the time on the secondary diversification. So once you have that VDJ rearrangement, then things can happen in immunoglobulin that cannot happen in a T-cell receptor. You get class switching, somatic hypermutation. Somatic hypermutation is when changes occur in that V region. Class switching is when changes occur in that C region. And there's something called gene conversion, which we're not going to spend much time on. Well, a whole chunk of that V region can actually be replaced. So you have your primary IgM, and this is IgM, where you then actually get a bunch of mutations in that V region that lead to greater diversity, somatic hypermutation. We'll see how that happens in just a second. And then the IgM can class switch, and it can change its constant region out. It splices it out, and it brings in something new. It can bring in IgA, or gamma can bring in the IgG. And then there's something, this gene conversion, which again is just not really covered in this chapter, and I don't think we need to know that very much, but a whole chunk of this can come out to lead to even more diversity. Both of these, so the somatic hypermutation class switching is mediated by something called AID, or activation-induced cytidine deaminase. So if this happens in the immunoglobulin variable regions, AID comes into contact, then you get something called somatic hypermutation. If AID comes into contact with the nucleotides in the switch region, then you get class switching. So how does this occur? So AID can only work on single-stranded DNA. But as this is being transcribed, as the DNA is being transcribed, it's going to be opened up, and it's going to, parts of it are going to kind of open into a bubble and become a single-stranded DNA. And this is when AID, depending if it's been turned on or not, can actually come in. And what it does is it attacks these cytidines, and it changes them into uridines. But remember, in DNA, all you should have is ACTG. RNA, those T's are substituted for U's, the uridines, but not in DNA. So this is considered foreign to DNA. So then your body's going to do something about it because it doesn't want those uridines there. So AID has changed the cysteines into uridines. And then two things can happen. So something called MSH26 can come in, and then once it detects these uridines, it repairs, and then it causes nearby mutations in AT pairs. And I don't know exactly how this happens, nor am I sure that it's that important. But the second thing that can happen is that these uridines can actually just be removed. Remember, your body doesn't want them there. Uridines aren't supposed to be in DNA. So comes along, and it takes those out. So then you have these abasic sites. There's no base there. There's no nucleotide there. So then this gets repaired using something called Rev1, where you actually get the mutation right at that CJ region. So all of these mechanisms can lead to the somatic hypermutation if this is in the variable region. If this is in the switch region, however, the same thing happens. A comes, and it changes the cysteines into uridines, and comes, and then it removes these uridines and makes them abasic. And then these abasic sites get nicked. Remember, this is single strand here, so you get a single strand nick using APE1. So then you actually have a break. You're going to have a bunch of these little nicks. And when this happens, you can actually get a class switching recombination. What do you think the consequence of not having AID and or a mutation AID and so it's a non-functional protein would be?
Hyper-IgM.
Hyper-IgM syndrome. Right. If you can't nick, then you can't class switch. So you're going to have a whole bunch of IgM. And probably, although I don't know this has been proven, you're not going to have as much variability. You're not going to have somatic hypermutation. So even those IgM you have probably aren't going to get as specific as in a person who does not have these mutations. So you're going to have hyper-IgM syndrome, a form of hyper-IgM syndrome. What are the other causes of hyper-IgM? CD something and CD something ligand. Number. Yeah, CD40 ligand, both called hyper-IgM, because that's the signal that's going to cause the switch. That's going to signal CD40 ligand is going to signal all the way to the DNA cause all this to start. But a problem with any of them will cause hyper-IgM syndrome. Okay. Right, so once again, this is what happens. You have your DNA. It's going to start the process of transcription, so it has to unwind. It actually becomes single-stranded. And once it's single-stranded, AID exploits it. It comes in and attacks all these Cs. That makes it into U's. U's shouldn't be there. This was the switch region, or either region. UNG comes and makes it abasic, clips it out, and then APE has the opportunity to actually nick the DNA. So this happens in these particular regions, and I don't know if we know why, more than anywhere else in the V regions. And these lead to your CDR1 and your CDR2 and your CDR3. These are going to have a lot of mutations that occur by this process. And I don't know if these regions just happen to be rich in Cs, probably.
The TV remote is going to cut off on you.
What's that?
Turn up the volume on the TV remote, or the screen's going to go blank.
Oh, I see.
That was a little warning at the bottom of the screen.
Okay. Right. So again, these regions are going to become even more variable as time goes on. Right, in the lymph node, in that germinal center, it's encountered antigen. That AID is turned on, transcription is occurring, and you can get all these mutations, somatic hypermutation. Somatic hypermutation leads to what? It leads to affinity maturation. So you're maturing, your antibody is maturing to have a higher affinity for the antigens trying to attack. Not every mutation is going to result in higher affinity. Some are not. But if it gets worse, then what's going to happen? Well, you're probably not going to keep signaling to the nucleus, so that cell's just going to die. It's not going to, those signals that are happening are also signaling for this cell to survive. But if it's getting a better affinity, well, that signal is going to be even stronger, and those cells are going to be selected to live. So you're getting a higher affinity. Somatic hypermutation leads to affinity maturation. So this is just one more of the same thing, right? You have your BDJ, and then of course initially you're going to have mu and delta. But then when you want one of these downstream, transcription is going to start. AID, and then followed by UNG and APE1 are going to come through, and it's going to nick. And when it nicks, right, what needs to happen? Well, your body senses your DNA has been broken. So your DNA repair enzymes are going to come in. And one of the DNA repair enzymes that's thought to occur in this is something called POL-nu. POL and then Greek letter nu. That's the repair enzyme that's going to repair this and bring it back together. So that's actually all. I skimmed the next section, which is evolution of adaptive immune response, and I didn't think any of it was actually worth spending too much time on, so I'm not going to cover it. So next week, I believe, we're going to do Chapter 6, Antigen Presentation to T lymphocyte. I think that's the last chapter before Christmas, if I remember correctly. Let's see. Stuff early on. So get a start on it. The only other thing I wanted to show you, just to kind of I mean, particularly in this time when we can sequence DNA so quickly, it's really behoove you to get comfortable thinking about DNA again. I just want to show you. So we always think of, right, when we think of the NIH resources, PubMed is the first one that comes to mind. But actually, if you come to PubMed, there's all these different things that you can use. And a lot of these, like, right, different taxonomies, they all, you can read about them, have fantastic purposes. But you can take one like a nucleotide. Or RefSeq, where you can actually find a sequence that you're looking for. So, right, if I wanted to find RAG, and you wanted to take a look at the sequence, so I always immediately change it to just Homo sapiens, because you're going to get a whole bunch, 2,000 different sequences that involve RAG. Change it to Homo sapiens, it gets much smaller. And then the trick I know, and I don't know why, the session number is always going to start with an NM, so you kind of have to search through and find what you want. So you're looking for an NM. These don't seem quite right. Looking for an NM, RAG. Let's see, found this. Second page. NM, okay, RAG. Oh, well, here we go. This one is actually called RAG2. I care about this. So if you click on it, it can tell you who sequenced this, how many times, what the references are, and then it will actually come down and tell you what the translation is. But then here is actually the whole, you just copy and paste this, the whole sequence of events. If you look fast, I think we'll just bring up the sequence. So if you're looking for a particular short sequence, you can actually just do a control find and, you know, make it up. Quickly find that sequence. So you can actually, you know, very easily pull this up. You can send it to your clipboard and put it into some different software like BLAST that's associated with free. There's lots of freeware. If you want to analyze this better, you can actually take it under protein. I'm forgetting what's called. The NIH actually has software. You can bring it down. You can take this, and it will, based on the characteristics of each of these nucleotides, fold it for you and give the theoretical folding, and it'll show you the crystal structure. And then you can compare it to a crystal structure that, well, I guess it's not a crystal structure, but it'll show you the folding pattern of the protein, and then you can compare it to a crystal structure that's been done, all in the same database. So again, you know, every time, once in a while, when you think about a gene, it's probably a good practice to just call it up in the NIH database and start looking at it as often as you do PubMed. I really think in the next 5 to 10 years, especially in our field with immunology, stuff is going to become very important to just kind of jog your memory and remember what point mutations are. And something that is actually fairly fun to do is you can actually then, okay, so you can take this and cut and paste it into a folding program and see what it should look like. And you can actually do a prediction if you have a novel mutation in a patient who has a form of SCID, and it will refold it for you, and it can show you how messed up it can then actually get. And those predictions are, again, based on, right, so if you have a 3 base pairs, right, that doesn't change the codon, so it doesn't change what the translation is going to be. It's going to be the same amino acid. Well, best we can guess right now, maybe that doesn't make that much of a difference because the protein is going to be the same anyway. But if it changes the amino acid, then the amino acid has different characteristics, like you know, whether it's hydrophobic or not, hydrophilic, and if it has a charge. And based on that, it will predict how it's going to change the folding. You can actually look, and that's how people kind of get a sense. And when we sequence, we kind of do this whole genome sequencing of individuals where we're looking to see where it is. Well, if we find something suspicious, then we can actually put in this program and refold it and say, oh, well, this is probably pathogenic, and then you maybe call a colleague at an institution who has a mouse model or something of that and say, hey, if you get this, what happens? Have yourself a nice paper. All right, any questions?
I was trying to understand the radiation sensitivity concept.
Mm-hmm.
I remember one of our patients, I think it was the last one, we had RAG1, oh, we had Artemis.
Yep.
So radiation sensitive just basically means like when you have exposure to radiation, you have breaks in the DNA, and the enzymes are deficient as far as repairing.
Correct. And those are ubiquitous DNA enzymes. So they're the DNA enzymes that are found in all your cells.
So like the DNA ligase or like different
Exactly.
ones? Not
Artemis.
Okay.
So RAG is the one that's specific for lymphocytes,
Okay.
and it's not in all your other cells.
Okay.
So RAG is not radiation sensitive, right, because that's just in your lymphocytes. But all those other ones, right, DNA ligase, I don't know, does anyone have a mutation TPDT that's pathogenic? Artemis, right, so those are the radiation-sensitive ones because you're going to have problems repairing DNA in all your cells if you undergo radiation.
There's one disease that's strongly associated with poor enzyme repair. Ataxia telangiectasia.
Oh, right. That was I can't remember what it was. Yeah.
Right, but there, I'm sure you get an increase in cancers and all kinds of other stuff too.
And so when you say, okay, they're radiosensitive, that means that they specifically have a defect in one of the ubiquitous DNA repair enzymes.
Yep. Okay. Like ataxia telangiectasia gene repair enzyme.
So then they were looking, when we had that Artemis patient, we were looking at further testing to see if they're really radiosensitive. What testing are we looking
how do you
for then?
test for it?
Well, how sensitive they are depending on the mutation, I have no idea. I don't know. I wonder if there's a test where you can actually take some cells from the body and radiate them and see how viable they are afterwards. I don't know.
Yeah, people have
I've never run that.
People have been measuring repair enzymes for 20 years.
Yeah, it was weird.
Some
They were.
stage or other.
He's he's already on the CMT service, and they were kind of looking into it, but they wanted us to look into it. And I remember I don't know, I'm having a hard time finding like what assay you would actually order.
Yeah, no, I don't know.
But
I guess it would depend. I mean, my initial thought is, well, if it's a problem Artemis, well, it is, but maybe depending on where the mutation is, maybe it's more or less sensitive or something.
Right.
I mean, that makes sense to me.
Yeah.
No, I don't know much about it. Thank you, Meg. All right.
Cool. Thanks.